welcome to the first of a series of JLS podcasts on leaders in biotech industry. Our first interview is with Jonathan Sporn, CEO of Gilgamesh Pharmaceuticals, easily the leading psychedelic firm globally with the recent interest by pharmaceutical giant AbbVie. Jonathan is a former Pfizer exec, Harvard MD, extremely experienced in the field, and a great privilege to have Jonathan here today. And of course, for our audience, this is not Jonathan's first rodeo. He brought many drugs to market as a high executive for Pfizer over 20 years, and also formed Perception Neuroscience, am I incorrect here, and which was spun out to Atai some years ago. Some interesting details for our audience. Gilgamesh Pharmaceuticals was founded 4 years ago with a $5,000 investment, and then grew through Series A rounds at 80 million. Then NIH came in with, am I wrong here, Jonathan, 13 million just 5 months ago.
The NIH doesn't take stakes in companies or anything like that. So, they just provide, non-dilutive grants and that sort of thing.
Well, non-dilutive is very nice when it's around $13 million or so. And then AbbVie came in with, 60 million in cash and 2 billion in milestone payments, easily the largest transaction in psychedelic history.
Yeah.
Maybe you could tell us a little of your origin story, Jonathan. I recall you partnering with Dolly Sames, the chair of chemistry at Columbia, and working in a little room next door for 6 months on your Mac, designing Gilgamesh Pharmaceuticals. Could you tell us about that?
After I had left Pfizer and started Perception, and then had sold that off to Atai, I decided to start a fresh company that would really design novel molecules that are kind of inspired or sort of related to things like psychedelic drugs and ketamine and those sorts of targets. So I had a very close friend who had been in charge of a lot of the external facing activities at Johnson & Johnson, had invited me to his lake house north of New York with a young chemist who was very entrepreneurial named Andrew Krugel, and who worked for Professor Dolly Sames at Columbia as a postdoc at that point. And so we sort of hit it off, and then I realized, as I was leaving the Thai folks behind, that these chemists that Dolly and Andrew and such were pretty unusual characters in that they were very creative chemists, and they really knew the space in the sort of psychedelic realm. So we got together about that time that I had exited from the Atai people, and I called Andrew up and said that I thought we should start a company, and so Andrew and Dolly and I had and I think Mike Cunningham maybe as well. We all had dinner in around the Columbia campus, and began to sort of map out how we would do this, and so that's how it got started.
And were you actually in the little room at Dolly's lab? He pointed out a little room next to the postdocs where all the fume hoods and glassware and what have you, beyond the locked DEA safe in the back room.
We were licensing some things, particularly the Ibogaine analogs from Columbia. So and everybody was there at that point. So so we did spend a good amount of time back in those days, around Dolly's office, and before we kind of started the company in earnest.
Just out of curiosity, how do you manage to acquire essentially the staff of a university laboratory into a private enterprise? What's the arrangement with universities generally in the situations like this?
Oh, well, I mean, most of what Gilgamesh does doesn't have anything to do with Columbia, other than, obviously we, are found, a lot of our founders are from Columbia, but mostly you're pretty careful about separating those things out. So so in this case, there was as I mentioned, the licensing from Columbia of certain asset group, and that is to everybody's advantage, although it can be kind of complicated getting the licensing agreement with universities done, but then that's to everybody's advantage, and then we try to be very careful about making sure that then anything else we are doing in terms of inventions and such are squarely being done by just by the Gilgamesh employees, and making sure we're not contaminating things by with the university.
So was that a licensing through, for example, the technology transfer office of intellectual?
Yeah, that's a pretty common thing. That license, led to the later to our applying to the NIH for funding for that program. So it was that whole Ibogaine space is super interesting, but it's also risky. So it was, I think, kind of one of an ideal place to have the government pitch in.
So your portfolio, if you will, your research directions for Gilgamesh involve non-hallucinatory psychedelics, and also Ibogaine analogs, and other molecules that somehow don't fit into either category. Could you describe the current directions, your hopes for Gilgamesh, the various directions that Gilgamesh is headed, research-wise and product development-wise?
Yeah, no, we're placing a number of bets, and in kind of a variety of directions, and so we were, I think, from the beginning thinking about it that way. So we have right now in the clinic one molecule that is a classic 5-HT2A psychedelic drug, and that we've developed just to essentially make a molecule that has strong intellectual property and is differentiated so that it is able to give you a very powerful psychedelic experience, but you can ideally get people within an hour or two in and out of a clinic setting. So that was the goal of a program like that. So that's on in the psychedelic direction and then we also, as you mentioned, we have a program with AbbVie that's called the neuroplastogen program, which is essentially looking to create molecules through a variety of strategies that are able to get this same kind of rapid acting anti-anxiety or antidepressant kind of activity, and yet minimize or eliminate the trippiness or the psychomimetic experience from it, and so that's of course controversial, and the proof of that this strategy will work, it's going to take some time to play out, but it does sort of contrast then with the kind of drug where you have to give it to people in the clinic and do a lot of very careful monitoring of people. This would be much more on the lines of how drugs are distributed and kind of dealt with today, where you can pick them up at your pharmacy and take them at home and this sort of thing. So those are sort of two contrasting things, and then we have our and as ketamine and S-ketamine and these NMDA antagonists are very, very important in terms of this kind of new way of thinking about psychiatry, and so there too we've taken the approach of saying, well, having to put IVs in people and have them come to clinics all the time, or same thing with nasal sprayers, is not, we didn't think, at least ideal, but we were convinced, as with our psychedelic molecule, that the precedent has been set that in this case with ketamine analogs, that they're effective, rapidly acting and effective, and have sizable effect sizes. And in this case, we created a molecule that's orally active, and that we think will have fairly minimal dissociative effects at the doses that we're giving it to people in this depression space that we're starting that compound out at. So we're there again, sort of creating a new molecule that will be a simple ketamine analog pill that's safer. And so we think that that's another sort of compound, sort of target product profile that is probably attractive to people if we're able to pull that off. And that's a couple of these studies are in the clinic and finishing phase 2 studies by the end of the year, or by the first quarter in any event, somewhere in that, depending on how you define finishing, because these things have a few parts to them. But so that's the sort of thing we're doing. And then we have the Ibogaine program I mentioned that's the NIH has fully funded and that's more in the substance abuse world, and that we expect to be very psychedelic, as Ibogaine itself is, and there we've managed to create another target product profile that we hope will accentuate the efficacy and pharmacology of Ibogaine, but remove some of the safety challenges that you have with that molecule, such as cardiac toxicity in particular. So that's the idea. And then we're bringing now I think as the company's now more kind of more funded, better funded, and kind of moving along, I expect that it's likely that we'll see over the next year or two additional drug targets and molecules that are going to move forward as well, so that we're trying to develop a fairly robust drug pipeline in neuroscience here that's beyond just, so we're not a kind of one drug or two drug kind of company, but something that I think will hopefully lead the field.
That's an extraordinarily diverse and indeed robust pipeline. You have essentially 4 directions. You have the strong 5-HT2A psychedelic. You have the AbbVie neuroplastogen track. You have the S-ketamine, MDMA antagonist analog track, and you have the Ibogaine variants for reduced cardiotoxicity. That's a broad and vibrant, requiring lots of postdocs and funding. Let me ask you a question about each one of these very briefly. In the 5-HT2A psychedelic track, the strong psychedelic track, the classical approach, if you will, what's the duration of your lead compound and dosage of your lead compound?
The compound has a half-life of about 45 minutes, which contrasts with, say, psilocin, which would be about 2.5 hours or so. And so the pharmacodynamic, that is the length of the sort of experience, is somewhere in the hour to hour and a half sort of time frame, depending, of course, on the dose you give people.
And what sort of dose are we talking about here? Is it 20 milligrams or 20 micrograms, or where are we?
Yeah, so it's we're still working on exactly what doses we'll target in late stage development, but yeah, these are in the kind of 5 to 15 milligram kind of range of doses we'll probably be administering, in the trials over time.
Do you have an ADMET profile or any type of safety or toxicity observations on this molecule at this stage? Any animal work?
Yeah, well, of course, as to get any of these things into humans, you have to go through a couple of small and larger animal species to, make sure that it's safe enough. And so the nice thing is these molecules at these sort of doses are, very safe. The kinds of adverse events that you mostly have are the psychological experiences people have with psychedelic drugs, and because this is lasting an hour or so, it's very self-limited. So even if someone were to have a difficult experience, they're over, within 2 hours, they can head home.
Oh, well, home is the place to be, I understand. I'm glad to hear that. On your program with AbbVie for neuroplastogen, I could ask the same questions. Is this a variety of compounds that you're looking at? And are these being generated on the bench by, say, Columbia postdocs?
No, it doesn't have, no, we don't, we don't really, have any laboratory or discovery work that's connected to Columbia for the reasons stated previously. So everything is, all these molecules are being designed as we speak, or being designed within the Gilgamesh chemistry and biology group, and then basically put through a testing funnel that looks at both safety and efficacy signals, and looks at their metabolism and all kinds of things that basically check boxes in this kind of systematic way.
Yes, Jonathan, using the interesting term neuroplastogen, I just wonder, one, how do you manage neuroplasticity? And two, what are the outcomes that you expect from, say, a compound with heightened neuroplastogen properties?
We needed just a term to use to, describe this sort of kind of novel area, and so that's kind of connects it in some fashion to what we've learned from psychedelics, but also distinguishes them because we expect that they won't be psychedelic. So how we do that is, I mean, I think the core principle here is mainly that you're looking at drugs, whether they're psychedelic or neuroplastogens, that are demonstrating this efficacy signal that is persistent, initially in animals and then in humans, far beyond the drug being in the body or sitting on the receptor. So the drug's been, administered, metabolized, but there's a change in the neural circuitry, for that individual, such that even in the absence of the drug, they're still experiencing a positive effect, on things like mood and anxiety. So I think that certainly, in the kind of academic world, people are now, starting to use fMRI and such to demonstrate that these drugs are shifting the neural circuitry of the human brain far beyond far beyond where the drug is on board, so that you can actually start to see how cortical hippocampal interactions, for example, are changed even weeks after the drug's on board. So in theory, those things are possible to do. In practice, we just want to show that these drugs work when administered on a kind of intermittent basis.
Well, Jonathan, if you're looking at post-administration neuroplastogenicity over a period of weeks or months, say in the cortical hippocampus, then you may be affecting more things than simply anxiety. You may be affecting establishing a reorganization of neuroanatomical structures that perhaps could have influenced training or learning and memory. Can you address the possibility of neuroplastogens being used for restructuring personality, reorganizing one's ability to absorb and assess data?
Yeah, all these are sort of, kind of, fascinating areas, and could lead to, the potential for these drugs to be used, beyond where we're at now, which would be, say, depression, anxiety. I mean, if you think about things like post-traumatic stress disorder or borderline personality disorder, there's lots of things that one could imagine, and that these things could, eating disorders, that these things could, allow a sort of, a more profound change in the way that individuals are processing them, the world around them. So it's all possible. I mean, we're starting out, we're doing the kind of science that, leads these things to more less efficiently get developed so that they're on the market, and that's a regulatory science approach. And I think once that happens, and these things are actually more available, I think that there'll be a tremendous amount of additional innovation that goes on in terms of how you look at the drug, plus various kinds of behavioral inputs, to and how these things are used and what sorts of individuals and such. So I think yeah, we're obviously I think we're not a university or an academic institution, so we have to stay pretty focused. But I think that there is a vast opportunity here with a lot of these things.
So you're familiar, of course, with the work of Gould Dolan, now moved from Hopkins to Berkeley, and her window of opportunity, a post-psychedelic effect, which could be a few weeks or a month, post, if you will, the long night of the soul, the various psychedelics. And you're proposing that neuroplastogen has a non-hallucinatory component, but still has the possibility of reorganizing or restructuring with intention human personality, emotional states, feeding behavior, potentially learning new habits. Is this what you're suggesting? You'd like to expand on that?
That sort of whole concept that Gould Dolan had proposed, requires, I think, a considerable amount of confirmation and replication. But I think it is sort of interesting in the sense that it speaks to this idea that that there are sort of kind of windows, kind of neurodevelopmental windows that we know exist, for all kinds of things, that we understand from classic neuroscience, and so sort of applying that conceptually to kind of social emotional kinds of learning and these sorts of things that where one could potentially open those windows again, or prolong their staying open is interesting. So but I think it's it does create this tension right now, I think, within the whole space, because if we're talking about using the traditional classic treatments in psychiatry, they're mainly symptomatic treatments, and if you stop taking your drug for attention deficit disorder, or your antidepressant, or your anxiolytic, you're probably going to be somewhat back where you started, or vulnerable to relapses of these kinds of symptomatology. So it's very different when we're at least conceptually thinking about drugs that may allow you to sort of be redirected into a kind of a developmental path that you were not on to start with, and I think that I think a lot of people probably are excited about psychedelic drugs because they've sort of, at least people that have experienced them, sometimes have not always, but sometimes have experienced that phenomenon where a episode with one of these drugs has sort of shifted where they're going in the world. And there's these classic examples, maybe even from things like Steve Jobs and Apple, where so these drugs kind of allow for something to click that allows them to then basically have a vision and a path to bring something forward, in themselves and in the world that's different from where they started, and these can be kind of rather dramatic transformations. So I think that's the kind of idea that we're talking about. And then it becomes very complicated when you're talking about drug development, because the FDA generally is regulating the development of pharmaceuticals or devices or these sorts of things, and less so, they're less comfortable with, and their mandate's not really for regulating psychotherapy and behavioral kinds of approaches so much. So I think as these things, as the sort of drug and kind of behavior modification of behavior and cognition, kind of move towards each other, I think it's creating some strains. And we saw that, I think, with the whole Lycos situation more recently, where I think there one amongst a number of tensions there was the FDA's somewhat discomfort with the fact that this was sort of developed as more a drug to enhance psychotherapy. And so the psychotherapy was more front and center in that particular instance, and I think that was sort of difficult for them. So I think there's a balancing act there right now, where companies want to get these drugs approved so they're out there and people can then innovate around them. But they also have to be thinking a little bit about the psychological support that is kind of part of the making sure people have the right setting when they're given the drugs and these sorts of things.
Well, beautifully stated, Jonathan, and profound implications for neuroplastogens. On your third track, the esketamine NMDA antagonist, you spoke about reducing the toxicity, the potential. Would you include, let me restate that, would you include addiction proclivity in that? We're seeing, along with J&J's remarkable profitability in terms of ketamine, widespread addiction syndromes. Everyone knows a friend who's perhaps using ketamine on a daily basis. Would these new compounds that Gilgamesh is developing have reduced addiction liability, or provide insight into underpinnings?
I mean, obviously, all of this is subject to, creating a full set of data that, will then be, evaluated by the DEA and subject to scheduling levels, but I think that there's certainly the reasonable expectation here that although I think a lot of these drugs will end up scheduled at some level, just because just the FDA is a very conservative organization. I think we should be in a situation where with drugs with very minimal dissociative effects, I think those drive some of that abuse potential. I think with drugs that really have don't have any binding to opiate receptors, for example, esketamine has more has some binding to mu-opioid receptors we don't tend to see with our compound. Things like that, I think we'll probably see that, even though we won't eliminate all abuse risk, that it will be relatively smaller and manageable, in the context of patients where we're treating people with for depression, and I think there'll be a lot of ways to manage that are not that. So I think it'll be challenging, but I think the efficacy of these things will be so much better than existing molecules that they'll be a strong rationale for managing whatever the risk remains.
Well, that's the progress in pharmacology. I guess here you're positing that S-ketamine has some binding with the mu-opioid receptor, and that underlies some of the addiction that we're seeing, and that your compounds have less binding with the mu-opioid and therefore, and less dissociative effects. So you expect fewer problems with it. The question is, will the compounds have equal or greater efficacy in treatment of depression?
Yeah, these are all molecules that are channel blockers at the NMDA receptor, and I think we're pretty confident at this point that this is a precedented target with demonstrated efficacy, because that's usually the problem in neuroscience. Is you start with some new target, and the chances that it's going to be safe and effective are very small. And once you get to the point where ketamine, S-ketamine, where these things have been out and around, and are have approved indications for S-ketamine and are just being used, become kind of established clinical treatments, with ketamine, you change that probability of success, pretty dramatically. So we think that there's it's not done till it's done, but there's a strong probability these will have a significant and rapid acting efficacy.
That's certainly praiseworthy. Jonathan, your fourth track, and for many, the most intriguing and controversial, is the attempt to create Ibogaine analogs for treatment of addiction, treatment of trauma. The many varieties in which Ibogaine has been applied: meth and cocaine addiction, alcoholism, fentanyl addiction, this sort of thing, but with a worrisome effect of the cardiotoxicity and the ensuing deaths from it. Can you expand on the Ibogaine work from its inception to its current stage, and what your hopes are for that?
Yeah, I mean, essentially, Dolly Samish and Andrew and company had been working on these things for like a decade, and so a lot of resources went into kind of creating libraries of molecules with what is a very complex pharmacology that involves sort of partial agonism at kappa opioid receptors and activity in NMDA receptors and such. So I think that what we really have placed a bet on here is that that essentially these molecules, that in animals anyway, they look efficacious in terms of things like if you have rodents that will work to receive opiates, they'll lever press to get a shot of opiate, because they're pleasurable drugs. You can reduce that sort of work for opiates by pre-administering these kinds of Ibogaine-related compounds. So we think that that sort of evidence and evidence that the withdrawal symptoms, that if you addict rodents to opiates, you then withdraw them, the withdrawal symptomatology is much less. So those sorts of things that it's hard to know how that entirely translates to humans with a more complex addiction problem. But at least that sort of evidence gives you encouragement that these new molecules we're making may perform as well as Ibogaine on efficacy. And then And then what we also see is that if you take human heart cells, human cardiomyocytes, you can show these electrical abnormalities that you see with Ibogaine. But with these new molecules that we're looking at, you don't see that abnormality, and that's in human heart cells. So we have some some evidence that would suggest that on both the efficacy and the safety front, these are at least promising. But now we need to get the particular molecule we're developing with the help of NIH through all the safety animal work and do all the manufacturing and such. So it'll probably take us another year or so to bring that particular molecule into human study.
It was my privilege to visit Dolly's lab, and one of the seminar rooms, observed Dolly rotating a stick and ball model of the Ibogaine molecule. And the question is, with these new materials, are they based on the Ibogaine scaffold or moiety, or are they entirely de novo?
Oh yeah, these are molecules that are closely related. So they're based on the scaffold of Ibogaine, and I think what Dolly's described is that Ibogaine's a very interesting molecule, and if you can make very little changes in that molecule, and that kind of can have big impacts on the molecule's signaling. So these are all sort of, so far anyway, variations on a theme on the Ibogaine molecule, which is sort of it's a very complex pharmacology. So we don't really it would be a little bit hard to make molecules that are far off from that because because of how complex it is.
So here in New Mexico and a number of other states, there are policy groups, activists moving to engage the legislatures into diverting, if you will, opioid money into treatment using Ibogaine. There have been hearings in Kentucky, Ohio. There may be hearings here. At the same time, there are competing groups doing the same thing regarding psilocybin as a treatment for addiction. Personally, from my limited understanding of the literature, there has not been the body of work, certainly not the anecdotal support for psilocybin versus Ibogaine is more efficacious. Can you speak to that, especially in the light of yesterday's announcement of a substantial grant for the study of psilocybin for opioid treatment?
I think the evidence is just that things like psilocybin, for things like alcoholism are probably going to be important, and how you compare these things. Ibogaine sounds like it's probably more transformational in the substance abuse space, but it's hard. I think it's because of all these complexities of safety and efficacy, and also Ibogaine is very challenging experience that lasts a very, very long time. So that makes it more challenging as well. But I think it does appear like Ibogaine probably resets some of these dopaminergic systems more acutely than other things. So it's hard to know.
Well, I'd have to agree with you. The anecdotal support for so many years from Ibogaine for heroin addiction has been profound and extensive. Of course, the hesitancy of people to employ Ibogaine, there's a fear of the drug as extraordinarily deep. And of course, the duration of it and the toxicity has caused quite a few people to avoid that. Where psilocybin maybe seem more applicable to neuroses or certain emotional states, and certainly an easier and shorter experience. So I think there's room for both compounds. But if you're talking about a deep addiction, perhaps Ibogaine may be.
Yeah, and whether that's the case for all addictions or, and of course, an Ibogaine now has, people interested in it for other things like traumatic brain injury and PTSD and such. So, we think that that's another area where, government funding would probably be warranted.
Which raises an interesting point. Of course, you're familiar with Nolan Williams' paper in Nature Medicine in January, in which he treated a number of warfighters for traumatic brain injury, and they were sent to MBO Biosciences in Tijuana. But before and after their experiences at MBO, there were psychometric testing by Nolan at Stanford, and some of the observations that he observed were a form of cognitive enhancement, if you will, of certain improvement in attention and focus, significant difference. Can you address that, the potential for a form of cognitive enhancement from Ibogaine analogs in terms of improved cognition, cognitive processing, performance on various psychometric tests?
It's not an area I know that much about. I think that it certainly was impressive that that you could take people that had all these blast related kinds of injuries that were having were part and parcel of their having a lot of psychological symptoms, and that you could have such a robust improvement suggests that there is some substantial neuroplasticity effects of Ibogaine that may allow for healing of some of these what are likely, white matter tract dysfunction in people that is related to those blast injuries. But yeah, so and it's hard to study some of this stuff in animals because animal brains are not the kind of damage you get from these sort of things is a little different in how they manifest in animals. So I think it's interesting but so all of these things speak to the fact that this whole area is intriguing, and there are things popping up that are where the effects in this case of Ibogaine in terms of people with traumatic brain injury and psychiatric symptoms, the effects are so what's seemingly large that you almost don't need a placebo group, and sort of like you
that was the problem in the paper. There was no control group, and there were a number of critiques about it. But again, Nolan suggested the effects were so significant as to
so you get things very, very big effects. The placebo becomes like but you just don't see that kind of effect size from placebo. So you need some placebo control, but at least as a starting point, it's hard to dismiss.
Do you have any comments on the possible expansion into U.S. clinics? I think Nolan established Oh, with researchers at Stanford, Soniera, if you will, pharmaceuticals for Ibogaine clinics in the United States. Do you think, can you comment on that?
I don't really know much about the clinic business, so I don't have that much to say about it. But except that, as you mentioned, people are trying to get some public funds to help with establishing some of this, I would think that it's challenging because you're dealing with molecules that are not FDA approved, and so I don't really you may know more about it than I do. It's a little hard to if you're trying to get research data to support approval, you have the challenges of getting a drug approved where there's a substantial risk that needs to be managed in terms of cardiac safety. And, even if that's manageable, it's still a challenge to make sure it's all done right and you're doing it in settings where you have all of the proper personnel and tools to manage it. So it's not, it's not
Well,
simple.
I understand. Jonathan, of course, Gilgamesh is following the classical, rigorous, entirely United States-based approach to FDA approval. As a former Pfizer exec, of course, you're following it very carefully. But Nolan's paper introduced an interesting concept, which was if the people were not treated with Ibogaine, for example, at Stanford, which would be illegal, but were simply tested for effects on cognition prior and after to application by volunteers of this on offshore jurisdiction with less regulation. That was an interesting approach, and that opens the door to testing of many molecules by volunteers offshore, less regulated districts, and evaluation of the effects as they come back in the United States. Do you see that as a viable method of collecting more human data quickly?
You should be able to, for research studies, get an IND from the FDA or get a CTA from regulators overseas or from MHRA. So you should be able to do it anywhere. It's just more a question of do you provide the right safety data that allows the regulators, and are you able to make the drug in a good manufacturing product way so that people are comfortable with it in every respect. So it seems sort of like an unusual sort of one-off to me, more than a kind of way to process things in general. I mean, just if people are serious about it and they want these things done, for most things, you probably want to do the animal testing and get an IND and test the damn thing.
Your classical approach, then. Question: we see recently that Dolly was doing a raise for establishing a Ibogaine foundation. Perhaps I'm unclear on this. I don't have much data. Can you speak to that, or is that?
Yeah, I don't think there's that much to say. I mean, I think he's very passionate about the whole Ibogaine space and just this whole, the potentiality of that molecule, having, you can mine that scaffold for a lot of therapeutic value, and so I think so I think he's interested in being able to attract funds to Colombia that are maybe more difficult to get from the usual sources. So
This
So
is more philanthropic, if you will, donor advisor situation.
yeah, I think he's I think we're you know this is a classic sort of situation where you have very high risk, high reward sorts of things going on, and so it's not necessarily something that either big or small companies are going to necessarily invest in early on. So I think that's why NIH has stepped up to fund some of ours, and I think yeah, I think he'd like to sort of have a substantial operation that is within the within the university to do some of the basic work in this way.
So, so of these four tracks at Gilgamesh, AbbVie has the neuroplastogen track, which seems to be
Mhm.
lots of attention and enormous promise. Can you, are these tracks in the structure of Gilgamesh separable? Can you spin out, for example, your 5-HT2 psychedelic track? Can you spin out your S-ketamine?
Or it's not, yeah, or ketamine alec, but yeah, the only program so far we partnered is the neuroplastogen program with AbbVie. So everything else is to be determined.
And last question, Jonathan, and thank you for the lengthy, in-depth exchange today. Last question is, there's a revolution in pharmacology occurring with the development of AI. That's NVIDIA, benevolent AI, recursion. There's a massive change in the way drugs are being created and developed, almost in clinical trials. In silico, for example, as a platform known as Chemistry 42 that envisions molecules that are not human. They are machine generated. They are molecules from the vast chemical space that no mind of man has conceived. And they're taking these molecules to further AI platforms approaching, if you will, clinical work. So that a molecule that might take 10 years and $1 billion in the past is being developed in 18 months. And the question is, do you find these platforms perhaps applicable to some of the tracks of Gilgamesh down the road? Or will Gilgamesh, I assume, continue to use physics-based platforms where you're visualizing a three-dimensional molecule and looking at things like binding coefficients in the receptor? Do you anticipate Gilgamesh using sophisticated AI platforms, or will it remain in molecule by molecule every few weeks developmental stage? Can you talk about that?
We're using more, in silico. We do, in silico work where we are, looking at very large, vast libraries of molecules that are, then being docked at crystal structures that allow us to kind of be able to find new leads quickly. How much we'll end up using AI models, I really don't know at this point. It's kind of early on.
It is early on. Last question for you of the day, and thank you, Jonathan. Your patent, your IP is extensive and beautiful and detailed and highly sophisticated. I'm just wondering who writes all those marvelous patents with so many molecular structures and claims. And I mean, do you have a partnership with Wilson Sonsini, or who writes your patents? And
No, it's mostly the chemistry group. And we obviously have we have a group of patent attorneys that are supporting all of that, but it's really substantially driven by the chemistry group.
Jonathan, it's been a wonderful tour of Gilgamesh's thought world and the origins and potential futures of it. Let me just open the door to you and say, do you have any good final comments for our audience on the future of Gilgamesh or psychedelia or drug development or, you know, the
No, no, I think we covered a lot of ground already, so I think it's good. I think, I think everyone should just be looking out in the first quarter of the year, because there should be a lot of news coming out of the Gilgamesh world.
Well, we'll all stand by eagerly and listen for it. Your firm has grown from a conceptual stage a few years ago to $2 billion milestones, and is certainly, in my view, the leading and most promising psychedelic firm.
All right. Well, from your lips to God's ears, Leonard.
Well, thank you, Jonathan. Lovely to speak with you today.
Thanks for all.
Great fun. We'll see you again. Thanks.
Bye-bye.